Very happy to be here and to be able to share with you more of the background or behind the scenes of n-lorem, but then also give you additional information. Obviously, Dr. Bain yesterday talked about Sloan and Susanna, so I'm not going to go over that again, but I just wanted to give you more an overview of n-lorem, safety profile across all our programs, and then kind of like what we're going to do next, right? Where to go, how to expand. So basically just, this is the agenda over there. Sorry, it's weird not to have a screen behind me for once, but that's good. Just to remind everybody, in case people don't know n-lorem, so n-lorem is a nonprofit organization that develops antisense oligonucleotides for free and will provide those to the patients for life, or to the physician to give to the patients for life. This is really possible because the antisense technology has been known now for more than 30 years. There's companies who worked on really trying to understand the concept. There's products on the market right now that are treating patients, and so it's a validated technology. It's a technology that's also cost effective, so actually making ASOs is quite easy, or much, much cheaper, let's say, than making vector viruses. That's just the nature of it, and that's why it's also scalable. And so that's one of the reasons why we can do this, but the other reason is also that in 2021, the FDA put out guidance documents that clearly laid out the path of how you can go from, making an ASO to giving it to one patient. And so they developed these guidance. We're following these guidance to the T, but it has a reduced toxicology package, so we don't have to do studies in non-human primates as you would have to do for commercial trial. And so we do it in rodents. We still follow an extremely long process to go through safety, so doing in vitro tests in the lab, in the cells of the patients always, and then afterwards still doing safety tests in animals, but rodents much cheaper than monkeys. So that's one of the reasons why this is actually possible to be able to develop these drugs for patients, for one patient at a time. Just wanted to quickly give an overview of the process at n-lorem. So for a patient to be accepted at n-lorem, a physician has to submit the application on behalf of the family, and the physician has to be at an institution that is willing to treat the patient, and that will be able to do that. The way we look at it and the way we talk to physicians about this is really a collaboration between n-lorem and the institution and the physician. So n-lorem will cover the cost of developing the drug and making the drug and all of that. But unlike a commercial trial where physicians and institutions get paid for every visit that the patient comes in, we can't do that. We just don't have the fundraising capabilities to do it all at this point. I hope we did. And so once the drug is given to the institution has to find ways to fund that. So a lot of time there's philanthropic funds, research funds. There's also a lot of things within the trial that can be charged to insurance companies, and we're learning more and more. So we're trying to gather all that information and share with all investigators, but as you can imagine, it is different by institution, it's different by state, it depends on the insurance that the patients have. So there's a lot of information that we're trying to compile, but it's just coming in slower than I would want. So to give you an idea of how many patients or programs that we have, since 2020, when n-lorem was founded, we reviewed about 350 applications. So we know the need is there. KIF1A is obviously a nano-rare, or was considered a nano-rare disease then. I think it's not as nano-rare as we all have found out. There's more patients out there. But this was a way to get in there. And I think it's going to be true for a lot of the diseases we treat. They're nano-rare today. With more whole genome sequencing, we're going to have more patients out there. We have accepted 180 programs, so that we're kind of working on, and they're at different stages that we're working on. We actually are now treating 30 patients. So we have 30 patients in the clinic on an n-lorem ASO, different diseases, a lot of different diseases. And so since 2022, when we submitted our first IND, we have now submitted more than, or actually yesterday, we submitted our 32nd IND. So for people who are in pharma, they understand that 32 INDs in 2.5 years, that's just pretty good. But anyway, we're just not proud of that, but we're just happy that that also means actually more than 32 patients, because we've had INDs where we have more than one patient in there, so which is also, again, something that was new. Just to let you know, because I really do want to focus on safety and just kind of give everybody an idea of how we do this. So we have a lot of committees throughout the entire pipeline, I guess, or throughout the entire work, that really look at the work that we do and help us, make sure that we have the best ASO that we can, that we give to your patients. So we have an access to treatment committee. This is a committee that looks at every application that comes in. We look at, the different mutations. Is this mutation amenable to an ASO strategy? Not everything is, unfortunately. And so we just have to determine, can we help this patient? What's the phenotype of the patient? How are we going to track disease progression? We have a research management committee. That's the committee that once we've done all the in vitro tests, the tolerability tests in the rats, and we have to look at all the data, determine which is the best ASO to take forward for the GLP tox study. We then have a study treatment assessment committee. These are a committee of investigators that help us come up with the best treatment goals for each patient, for each protocol. Obviously, FDA is kind of like a big one that looks at all of our documents and makes sure that, they agree with the quality and everything in there. IRBs, every institutional IRB has to review and accept the protocol, approve it. And then we have a data safety monitoring board. So this is made up of external members, and these people look at the safety data on a quarterly basis of all the patients that we're treating right now. And so they will, look at all the adverse events, anything that happened, and let us know. And, if they think the benefit risk is still there and we should continue treatment. So again, currently, like I said, we're treating about 30 patients. Out of those, though, if you look at the 12 patients that we can evaluate, meaning they've received at least 4 doses, a minimum of 6 months of treatment, or they have had an assessment post-dose, because we obviously can only determine if there's benefit if we look at an assessment post-dose versus pre-dose. So we can say that out of 12, we have 11 that have actually shown some benefit. Right, so our ALS patients have seen their ALS score stabilize. They have seen the NFL actually go down, which is a biomarker to show that there's less axonal death. We see a lot of improvement in various different CNS domains. So we see improvements in communication in patients, improvement in behavior, so stereotype behavior reduction. We see improvement in stamina. We see improvement in neuropathy, motor, gait. So again, there's really a lot of patients where we see improvement. There's one patient we're treating with ALS where we haven't seen any improvement. This patient was extremely far progressed when they started treatment, but we still initiated the treatment. But to date, we haven't seen any progress. And we need to know that this is to be expected. There may be patients that are either too far progressed, maybe the ASO won't work as much, maybe there's going to be too many other symptoms that we just can't address with an intrathecal injection. All right, so focusing on the KIF1A ASO. I just wanted to go back on the handle thing and just going to run through this quickly because I'm sure Dr. Chung and Dr. Bain have talked about that yesterday, but you have 2 copies of a gene, so 2 messenger RNA copies. On one of the copies, your child has a mutation or you have a mutation, right? So that's the KIF1A mutation. Us humans, we also all have changes along the gene on either copy. We call those SNPs or handles, but I call them SNPs. So they're changes in the base pair. Basically, those where you see the X's on the graphs, it's just a change, from one gene to the other gene. And so we can use that to create an allele selective ASO, because what we want to do is we want to just knock down the one side, the mRNA that has the pathogenic mutation, right? We want to leave the other one alone because you do need that. So this program, unlike other programs that nlorem has also worked on, we can't just knock down the entire gene. It would be easier to design the ASO if we just knock down everything, but we can't do that. So that's why we have to look at all these SNPs. So then what we can do, we can use the fact that there's a change on that one side, and we can, screen ASOs across those. And so normally we can screen about 20 ASO per SNP that we can find. But we don't just have to focus on the SNPs that are on the pathogenic allele. We can focus on the SNP on the other side, because all you need is, you just need to make sure that the 2 genes don't have that same letter at that position, right? So we screened about 500 ASOs for that first ASO that's now in the clinic that's treating Susanna and Sloan. And so basically to show you how this works, so that benign SNP, that just letter change, is on the other side of where the pathogenic mutation is, on the other gene. So the ASO binds there. And And I didn't go over the mechanism of action of an ASO because I think Dr. Bain yesterday and Dr. Chang probably went over that. But basically, what happens is that the ASO binds to that messenger RNA. So ASOs are pieces of synthetic DNA that are just put together. They bind perfectly to that strand of the RNA. Our endogenous enzyme RNase H1 comes in because it detects an RNA-DNA duplex in the cells, and that's not supposed to be there. And so that enzyme comes to chew it out. And so then it kills the mutated RNA, so there's no mutated protein that's created. However, the other side just still creates perfectly healthy protein. So that's really how this ASO works. And so I think what was discussed before also, originally the drug for Susanna was created and we thought only Susanna would be able to use it until we did the whole genome sequence of Sloan a couple of years later, and we saw, lo and behold, she has the same SNP. There's actually, I think 50% of people just happen to have that SNP. And so it just needs to be on the right side. And that's why we need to do the long-read whole genome sequencing on every patient to be able to see, is it on the right side? Because you could imagine if it's on the side of the pathogenic mutation, we do not want to use it in that patient because we don't want to knock down the healthy one, right? It also means that for some patients who cannot use Susanna's ASO, they just enter nlorem's pipeline. So then we develop another one, ASO. And I'll go over that in a little bit. So I just want everybody to be confident that if you don't have the handle, that doesn't mean you can't get the treatment. It just means it's going to take more time for you to get the treatment. Also, having said that, even if a patient has the handle, and so everybody's like, yay, have the handle, I can come into the clinic tomorrow. Not really, because we are still doing the in vitro tests in the cells to make sure that the knockdown is the same as what we saw in Susanna's cells. Meaning we just still want to make sure that we have a 20-fold difference between knocking down the mutant versus the wild type. Obviously, with any drug, if you start pushing the dose high enough, you would likely knock down everything. But so we still need to do those tests, and those tests needs to be done in iPSC cells, and those take time to grow. So it can take 5 to 8 months to get an iPSC cell line. So I just want to put that out there for everybody, just so that mentally one doesn't get frustrated and say, well, I have the handle, I should get treated tomorrow, because we still need to do that test. Eventually, I would say maybe once we've treated or tested 20 patients or something, and we see that that knockdown is always the same, it's always the same fold selectivity between both, we may be able to bypass that. But for now, we just need to make sure from a safety perspective that we have that information. So in terms of the pioneer patient, just to kind of give you a timeline for people who like timelines. So we submitted the IND for Susanna in August of 2022. The FDA has 30 days to review any of those research investigational new drug applications. That's what IND stands for. So a month later, they came back with a may proceed letter. That's what they give you. They don't like to say that it's the IND has been approved. They're like, we haven't approved anything. We authorized you to proceed. So we have to use the right terms. So we got the may proceed letter. The IRB came back really quickly thereafter because we can do a pre-IRB review so that by the time we get the may proceed letter, we can just tell the IRB, please approve right now. And then on October 10th, 2022, she got the dose. And then for the second patient, we then submitted her application in February of 24. May proceed letter came March of 24. They always are 30 days to the dot, really. So, and then we got IRB approval really quickly and dosing really quickly. So this is kind of the path. A couple of things I just want to mention about nano-rare patients, and I think with the KIF1A, you are in a really good position, actually, but just wanted to say, nano-rare patients come to us, to all of us, physicians and researchers, with very unique characteristics, right? Every patient is different. Even patients with the same mutations can have a very different phenotype, as we know. And so they can also have quite advanced disease depending on delay of diagnosis, as we know. Hopefully with more whole genome sequencing, more awareness, people get diagnosed earlier, which is everybody's hope. There can be a lack of natural history data, and that's where you guys really have a foot up because of that koala study. There is a lot of natural history data, and so we have a lot more understanding of the disease, which is great. a lot of patients on multiple concomitant medications. Oftentimes people on medications to try to treat the symptoms, right? Because that's really all you have. And so you may have a lot of medications, and this is something we just need to take into account. We do not want ever anybody to get off any medication before they get an ASO treatment, but I think we just need to monitor how the meds are changed over time, right? We have had patients that have been able to come off some of the anti-seizure meds because the ASO is doing the work. So that can be good. That could actually be an efficacy endpoint at points. And then, patients will experience adverse events, and I'll go over what I mean with adverse events, right? But people will experience things while on trial. And so we have to figure out, is it due to the drug or is it just disease related? And also for a lot of these diseases, we don't know how the disease will progress with age. People will go, children will go through puberty, things will change in patients' life, and sometimes that can just change the phenotype a little bit, right? And then we also don't know, an ASO, for example, it's given intrathecally, it's going to the brain, it will affect everything that the brain affects, but there's gonna be systemic symptoms that we probably won't be affecting because the drug doesn't go to the rest of the body, right? So we all just need to be aware of kind of like those limitations and those things. That's why we need to track safety really well, and we need to track clinical benefit or any changes in patients really well along the trial. So how do we monitor safety in an n-of-1 trial? Really how we do it in any other trial. And I ran multiple trials at Ionis Pharmaceuticals before, so it's really pretty much the same process, as stringent, right? But what I do want to go over is kind of explaining the terms, right? So what is an adverse event? Basically, we have to collect for the FDA and for ourselves. An adverse event is anything that happens to a patient after they started taking the investigational drug. So from that day on, everything that happens to them, anything bad or something that, is an adverse event. So if they run into a table and have a bruise, that will be reported, can be reported as an adverse event. If they fall and hit their head, that's an adverse event, right? Whether it's related to the drug or not related to the drug is at that point not important, you just have to note it. What's a serious adverse event? So that's a level above. That's also a regulatory term where we have to report to the FDA if the serious adverse event was related to the drug. So what a serious adverse event normally leads to hospitalization or leads to being, having treatment like a surgery or something. Again, it's a hospitalization. But for example, if a patient has an appendicitis and has to go to the hospital to get that removed, that's not necessarily ASO or any other drug related, but it's still a hospitalization. And so we still are collecting that information, right? So just kind of want to give that point of view. Adverse events and serious adverse events are also rated on two terms, severity and relatedness. So severity is mild, moderate, severe. Mild is something that does not affect your daily life. That's how it's, so you have a runny nose, it's mild because you can still do everything you do. Moderate is it has some impact on your daily life. You might have, hurt your foot or something and you're just limping around, but you can do most of what you can do. Severe is something that takes you out. You cannot, go on with your daily activities. So, if the flu takes you out completely for 3 days, that you could consider that severe. And then relatedness, we use 3 different levels for relatedness. That's what the FDA likes to have, not related, possibly related, or definitely related. So not related is if you can easily say this is not related, like 2 months after let's say the ASO was given, the child or the patient has a headache. Unlikely related, it's just a headache, right? So that just happens. Possibly related. If you give the LP injection, you can get lower back pain. That can be procedure, that could be ASO. I mean, it's hard to differentiate. And so a lot of times physicians will be on the conservative side, and we normally like people to be on the conservative side when it comes to safety, and we say it's possibly related to the drug and it's possibly related to the drug injection, right? And so, so that's how we record it. Something that's definitely related is if you stop the drug, the thing will go away. If you give the drug, it definitely comes back, right? And so it's terms that are not super clear, but normally it's temporally related to when the drug was given. And also not related if you have a completely different reason for why this was do, then that's also not related. And then again, common side effects of lumbar puncture. All our CNS drugs are given through lumbar puncture, so there will be side effects due to that. Lower back pain, headache, you can even have vomiting, nausea, dizziness. All of those things could be just the LP, or again, it could be the drug, and sometimes you can't differentiate that. And then you can have rare complications with lumbar punctures also that are, infections, bleeding, and things like that. But those are extremely rare, and really we haven't seen any of that at all in all our patients. And I just wanted to say that, as a whole, if between the nlorem ASOs and Ionis ASOs, and I'm just mentioning here the Ionis ones because we obviously come from the same cloth than Ionis, having a same founder, they really have been generally well tolerated in many different populations, right? And so this is also from data that you can look at from the clinical trials that Ionis ran. So there's all the safety data there. And then we are trying to put out as much and as quickly data on our patients so you can also see that, the information from our publications. So in general for the KIF1A program, the Data Safety Monitoring Board met last June. And so we reviewed the data of all the patients. Nothing was... No event actually across all our patients was deemed definitely related to ASO. We had some possibly related, as you can imagine, as I just mentioned. We do have one patient that has vomiting and fever that comes up after pretty much every dose now, and so we are really trying to better understand what is going on there in terms of cytokine levels and complement activation and things like that. But we also know that 46% of patients, and lucky that you guys have collected all that data with the natural history study, a lot of KIF1A patients do have issues with thermoregulation and will get fevers that are not necessarily related to anything. So just trying to tease out what's what, right? Especially because the second patient doesn't see that. So we kind of have these two different patients. But overall, I would say that for the KIF1A ASO, and I know that you saw videos yesterday, the benefit really right now is much, much higher than the risk, right? And so there's improvements in neuropathic pain, tremor, stamina, communication, and so forth. And so I think this also shows that, the brain is plastic. We have two different patients we're treating, one that was young, very early in her disease. Obviously you would expect good things to happen there. One that was, more advanced in her disease, and we're still seeing good things happen there. So I think we can all say that the brain is more plastic. I will never not treat a patient, even if people are telling me, but they're really late in disease. I'm like, but we don't know what we can do. So I think this is really something important here. And then I wanted to quickly go over lessons, what we've learned from Spinraza, because I think it is applicable, and I'm focusing on Spinraza not only because I was part of running those clinical trials, but also because it's an approved drug right now, and so we have a lot of information, but I'm sure Tamara would say that for Angelman, a lot of the same things are coming true. So we all know that if you don't treat the disease, well, that's not good, right? So especially SMA in the infants, it was really a devastating disease, right? The earlier we treat, the better the outcome. That's also not rocket science. I think we all realize that by now. And that little graph there just shows the motor milestones of the infants in the clinical study and all the infants who on Spinraza, a lot of them met their milestones of sitting and rolling and, being able to do that, while the patients who got a sham control could not. And just as a reminder for the Spinraza, we actually didn't inject placebo because that would just not be ethical at all. So they just did a sham procedure where they just poked the back of the patient, kept the patient in the other room for the same amount of time as the injection would take, just so that the parents would not know if the kid got the drug or not. What we also learned, though, from Spinraza is that, again, even the older patients who got treated, not just adolescents, but even adults who got treated, did see benefits, right? So again, I don't think it's ever too late to start treating a patient. And really, I think we can say that Spinraza transformed SMA in a big way, right? It was the first approved drug, but it also led to many other companies coming out and trying to do additional treatments. Because I'm not saying that the ASO is the one best thing out there, but it's really pretty good. But now there's multiple approved treatments. No treatment is a cure. an ASO has to be given on a quarterly basis, right? Because it works at your RNA level. RNA is being produced all the time, so you have to go back and make sure you fix that. So it's nothing that affects your DNA. But I also do want to mention, though, that even the AAV therapy that is approved for patients with spinal muscular atrophy, it was given to kids before they turned 6 because the drug can then cross the blood-brain barrier more easily. And it was hoped to be a one-and-done drug. Even a lot of those patients are actually now on Spinraza or another disease-modifying therapy. So I just want to be completely transparent because I think it's important for people to know. The AAV was doing great, but at some point patients were plateauing, and then people started to do the ASO treatment. I think what is really interesting with SMA is SMA used to be differentiated in different types based on the severity. So type 1 was the infants, type 2, type 3. But now that we have disease-modifying therapies, they actually got rid of the typing because it doesn't matter. If you have the mutation, you should take one of the disease-modifying therapies and it's going to work. So I think that is kind of interesting. And then also, obviously, what is really great is SMA is a recessive disease. So mom and dad have to carry the mutation. They're unaffected, so they don't know. And then they can have a child with the disease, right, because of that. But now, first of all, newborn screening is accepted in all 50 states for SMA. Which is great, so the babies can get tested and you can treat them earlier. But a lot of parents also do prenatal testing to see if they are carriers. All right, so just want to quickly go to what's next for KIF1A now. So nlorem is committed to treating as many patients as we can. The FDA guidance said individualized ASO treatments for a couple of patients. It's probably purposely vague on the number. Well, there's no limit of patients. We all know that there's more patients there than one, obviously, seeing everyone here. So to date, we've accepted 12 patients into our pipeline. 2 patients are dosed with the current ASO, as you saw yesterday. There's 6 additional patients accepted that can use that same ASO, so that's great. We mainly have to do the cell work on them, and then we can start treating. And 3 are actually about to treat. We have an approved IND, an approved IRB protocol, so we just need to get the drug to the site and everything. So we have another 4 patients in our program that cannot use the current ASO. So we're going through the process of developing an ASO for those patients. Between those patients, actually, we may be able to find one ASO that can treat 2, right? So obviously now when we're screening, we're using the long whole genome sequencing of all these patients in parallel, and we're looking where are the common SNPs, right? Because why would we do it one at a time if we now have the data and we can just try and push forward, right? And I think likely, that will be the case. Maybe if we have 5, 10 ASOs, we could treat everybody with KIF1A. So who knows? Yeah. And I think one thing that I want to mention here that Tamara was saying that's really important, I think it is great, and you guys should all look at potential commercial trials and commercial development of these ASOs. What I hope nlorem has done at a minimum, I mean, we've treated 2 patients, we're treating more, so it's not a minimum, but what we have done is we have de-risked the program for pharmaceutical companies, right? So the patient population size was small. Pharmaceutical companies cannot go after a small patient population size. That just doesn't work, doesn't make sense for them. But especially because of the risk. There's always risk that it's not going to work. We have de-risked the program now. We have shown that if you actually knock it down, you can induce benefit. That's one big hurdle for a commercial company. So now hopefully that has opened the path. So we're also very keen to help and expand ex-US, right? We understand that there's patients everywhere and patients should be able to get the drug. The one problem is, like I said, the FDA has a clear path. The rest of the world doesn't yet have a clear path. That doesn't mean we can't do it. It just means that will they accept a smaller toxicology package, right? We don't have the bandwidth to do non-human primate studies. That is just not in nlorem's future, right? So we are very happy to work with anybody, anywhere. What it takes is financial and operational support. And what I mean by that is we have to have a committed physician with a supportive institution. So, and I was talking to Dylan today. We've had a lot of discussions with people, and we are very happy to go into Europe, right? We have started discussions for the other ASOs, with physicians, and we're talking, do we do a named patient path, which is an easier regulatory path in Europe, or do we do a full clinical trial application? Honestly, today, I don't care which way we do, but we just need to do it, right? And so, but talking about, we may have patients here. All I need is a physician and a patient, and I need the physician to realize we are going to run our clinical trials in Europe or in Australia or wherever, just the same way we're running them in the US. It's a collaboration with us. We're developing the protocol together. Data is going to be entered in an electronic database that nlorem runs like we do for the others. We're tracking safety. It's going to the DSMB. We're just doing the same thing because I have a responsibility to all the patients. And so if anything bad were to happen in Europe, I have to announce it, tell it to the FDA. I have to let the physicians in the US know. So it's not that I'm just going to go give the ASO to somebody and be like, have fun, right? So as long as all of that is there, we're good, right? The caveat is, nlorem is 32 people. So by now we have submitted 1 IND per employee at nlorem, and so there's a limited bandwidth. And so that's why it is also collaboration. We need the people on the other side of whatever pond to help us, see what documents they need, but we are happy to work with them. And, it might just take some time because we have to prioritize, submitting INDs here, giving paperwork there and stuff like that. So, but we are willing to do that. So as a summary, I think the clinical experience, across nlorem makes me personally very proud that I know we can develop safe ASOs and we can track the safety and we can track the clinical benefit. Again, with KIF1A, we have a favorable benefit risk ratio on our hands here. We see clinical benefit in both patients. It's sustained, it's long-term. Will things change over time? Maybe, but that's why we're just going to keep on tracking the data. So we're committed to pursuing treatment for as many patients as we can through US research INDs, maybe commercial partnerships. We're very open to that as well, starting to talk to people, and then we'll expand one country at a time. I do want to thank a lot of our supporters, especially in the toxicology and manufacturing realm, because these CROs give us, let us do the work at reduced pricing because they believe in the mission. And so without, their support also in that in-kind donation, we probably wouldn't be able to do it. So I just really want to make sure to mention that. And then before we go to questions, I just want to do one plug for the nlorem NanoRare Colloquium that we're having in October, so October 20th and 21st. If you want to come celebrate my birthday with me on the 21st, please do. But really come and celebrate the NanoRare patients and the Pioneer patients. We're really trying to talk about the entire process, give insights into things. And in nlorem fashion, it's a free event, and it's virtual and in person. So hope to see everybody there. Thank you.
Thank you, Laurie. And for everybody, I think this is, like I said, a place where we have as many questions as answers. So we are going to shift our schedule a little bit. We're still going to take our break, but we're going to take questions until 11 so that people can share and get the information that they're looking for. So any questions in the crowd?
Hi there. Thanks so much. Yeah, our son is just 3, and we are interested in exploring everything we possibly can for him. He's kind of a milder variant, and I guess I'm just wondering, firstly, if it's less interesting to treat those kids with milder variants? No?
No, I mean, I would say, and I think it as long as we have outcome measures or we have measures that we can use to see improvement or benefit in the patient. And again, like I said, I think, Dr. Shang's koala study is helping us be able, to track what the normal course of the disease is even for a mild patient and then what treatment would do. So to me, there is not too mild of a person or too severe of a person. And normally if you go in the milder, I mean, you should be able I'm not saying to help more, but to
Right.
bring them closer to neurotypical or
Awesome. And then the biggest obstacle that we saw was potentially funding. We'd heard the number $200,000 floated at some point, like something like that.
so that part. So
I'm just wondering where that's going, and I've heard some of it might be insuranceable. I'm not sure.
yeah, so I think that amount is off. Can be more on the institutional side. So from the nlorem side, there is no funding needed, right? The ASOs will be funded for. I mean, we do fundraising just like every other organization, and we can move our programs along based on our funding. So there's no funding on the ASO. It's from the institution side. Oftentimes, there are some things at an institution that the institution cannot bill, and I just want to also be clear because I just want to give the institution. I mean, they're doing a lot, right? When they're doing a trial like this, and they're taking on a lot, but there are costs that cannot be submitted to insurance. Pharmacy storage costs that you can't just, put that. So there's going to be some things, or the cost of the study coordinator that calls everybody to set up the appointment, that puts the data in because I'm bothering her to put the data in because I need to see the data and things like that. Those are like people time things that cannot be billed to insurance because there's no path for that, right? So, and every institution is doing it differently. Some institutions have a lot of philanthropic funds that they can put towards that, or they may run multiple commercial clinical trials and can kind of use 5% time of a study coordinator or something. I don't know. I mean, I'm not saying people are doing that, but just, I think that'd be just fair. But so that part I can't tell you,
Sure.
right? And it also depends by institution what it costs. But I think, if you hear that number, it's probably like the utmost it would cost if nothing was submitted to insurance.
Understood. Thank you so much.
That's how I think I have understood it, but yeah.
We're going to take 2 questions from online. A couple of them are similar in theme about how can international patients outside of the U.S. Access the treatment, which I know you addressed in your slide, but it seems people are asking anyway.
Yeah. So, right. So what we have done, just because it is tricky, and it is the only thing with international is tricky because the FDA has a path, right? And that's the main thing, right? The FDA has a path, so we know if we do everything they ask us to do, we submit the IND, they like the data they see, they will approve, they will authorize to proceed. And we don't have that in Europe or anywhere right now. If it's an ASO that has been developed already for a person that we have an IND for in the US, we need the patient to find a physician, the physician to contact us, and then we can see if we can work with them. The physician has to be willing and able to do this under, we don't know what path, named patient path, a full clinical trial application. I mean, we'll do whatever, but that's the path, right? But we need a physician. And so unfortunately, I want to help every family, but without the institution and the physician, we can't do anything, right? We don't anymore, we used to, but we don't really accept patients traveling back and forth, right? First of all, not everybody can do that, so that's also not very fair and equitable. But if something happens, like safety-wise, and they go to their local physicians, we had a patient very originally, and nlorem did that, and getting the paperwork from that hospital there, because they don't really know what they're doing over in the US and stuff, was just mind-boggling, right? So right now, connect with us. We can see if we can maybe work to get the drug there. Again, I understand the frustration. I'm not just...
A second question that's been posted online is, if a child has a mutation found in only 31 children worldwide, would you synthesize a new ASO if the current one isn't compatible?
Yes.
Back to in-the-room questions.
Yeah. Hi.
Hi.
So my first question is, ASO stands for?
Sorry, I imagine that they all talked about it yesterday, so don't you hate when people have acronyms? Antisense oligonucleotide.
Okay, thanks.
It's just the name of those types of drugs, so that's why ASO comes out much more
My
easily.
second question is, when you talk about we cannot use or we cannot start use this technique called in other countries, but my question for Canada. So I know Canada is not the 51 state as Trump
Thank God.
pretend, but I know that we have many similar regulations.
Yeah.
So my question, is it possible to do it in Canada? And is there any Canadian patient who started taking this?
So we have started to talk with the Canadian authorities, and so we are kind of far along. So I do think Canada will be possible very soon.
Okay, thank you.
So but you know, again,
Yeah.
you know, we'll keep you posted. But
I
yes, I
understood.
do think Canada is going to probably be one of the earlier other countries where we can treat.
Okay, thank you. My last question is, how long time to see the effect of the treatment on the patients? 8 months, 6 months, 1 year?
No, I think for KIF1A, I think it depends by disease, and also in every patient, I didn't go through that, but in every patient we dose escalate, meaning we start with a low dose for the first dose and then we go up, right? Because we, again, just want to do it safely, right? So even if we have one patient where we're already at the 80 milligram dose, for example, we're not going to start the third patient at 80, we're still going to dose escalate. So with that, we have, in some cases, we've seen signs of benefit after 3 doses. So you know, and that's 4 months because the first 3 doses are given closer together. But I would say 6 to 12 months should be something where I would feel more confident that whatever we see is not wishful thinking, you know, or anything like that. I think, and then you really need also a visit where you have an assessment, right? Because, well, as a parent, you obviously will know or see things at home maybe faster, but just for us to kind of feel confident that like that is a real improvement. But again, I don't want to also minimize what people see at home, because I think it's very important to get those parent input, and we try to do a lot of, you know, parent questionnaires and patient questionnaires when we can, because I think it's very important to also see what's happening at home versus just at that clinic visit. We all know that's just a snapshot in time.
Thank you.
Yeah.
A question from online asks, the KIF1A subjects showed increased cognitive engagement. For any of your subjects to date, have you been able to demonstrate measurable cognitive outcomes?
Now they're picking my brain here. Measurable. I mean, definitely in communication, right? So we've seen increased communication on the ORCA. That is 5-point change, so would be considered clinically meaningful based on data that we've seen from Angelman. Yes, we have seen, you know, definitely communication, people being more aware, people being, you know, able to do better eye contact, and in other diseases also. So I would say yes.
And then another question from online asks, how can we find out if the eligibility status for the ASO treatment has changed for my child?
As long as the child has a mutation that's amenable to an ASO strategy, the eligibility wouldn't change. So the way that the protocols are written, because it's for the one patient right now still, and even if we include 3 patients in an IND, which we've done, the inclusion criteria are not like commercial clinical trials. It is, you know, has a mutation in this, is able and willing to do all the procedures, you know, in the opinion of the investigator. I think we have like 2, maybe at most 3 inclusion criteria. There's no other inclusion criteria.
Hi, hello. I have a couple questions just for you, but is there any insurance impact by a patient who does the ASO? I mean, does it impact their insurability? Do you know?
I don't think so. I don't know. You mean insurability in the future?
Yes, yeah.
I mean, it shouldn't. In the US with GINA, you shouldn't be able, I mean, because that would be the only reason I think you would be impacted, right? The fact that that there's a mutation and there's a genetic disease, but that you should be protected for against that. But I don't know. You haven't had any change in your insurance?
Coverage of other symptoms that are not ASO
Okay, so just to repeat to everybody, for everybody online, Luke just said that there's been no change in status of Susanna's insurance. Insurance has covered pretty much everything, and there's not been any impact on other things being covered for Luke that is not related to the trial.
Thank you. I had one other question
Yeah.
for you too. When you say you need a physician to submit, would it be a neurologist, geneticist?
Yeah, so that's a very good question. Thank you for asking. It would be the physician that will become the treating physician. And so it doesn't matter, I mean, because I know that clinical geneticists have been giving the treatments, neurologists have been giving the treatment, so it doesn't, the specialty to me doesn't matter. It just has to be the physician who will be responsible for the treatment. So, the treating physician. So sometimes the treating physician doesn't do the LP themselves because the anesthesiologist does that. So it's not the anesthesiologist who should submit. It should be the patient who's going to be, the physician who's going to be responsible for the patient.
Thank you. And then my last question is for KIF1A. Now that Emory and we have 4 new patients that are through n-lorem, I know we previously, some of us have submitted blood work to see if we were eligible for the ASO that was developed for Susanna, and came back we weren't. Will you guys be sending out any notification with the new patients that are going through this if they are eligible based on their ASO?
Before Dylan answers, I just want to say they still need the info from us, and it is still in the process. So we still have, many ASOs that we're running through. So it's only until we have the last GLP tox study where we know this is the ASO that's going in the first patient that we'll be able to say, this is the SNP, and then we could give that information. We would give that information to KIF1A org, and then everybody can, see who's eligible. Yeah, you'll get that information.
We have a couple questions from online. One of them is, how long can an ASO be taken for? What happens afterward? And do we know what happens if treatment were to stop for whatever reason?
So an ASO can be treated so far for a very long time. I think SMA children have been treated since 2010 or 11 is when the first trial was started. And so some of these patients have been on since then. So, and then we'll have to see, because I think that's the one program that's been out there for the longest. So for life is normally, the treatment is normally for life, right? And so I think what happens if you have to stop the ASO treatment, because it works at the RNA level, and like I said, you're going to keep producing RNA, and you're going to have to take it forever. And if you should stop taking it, likely, you'll have the disease come back.
Should we take an in-room question? I'm not prepared with the next one yet.
This may be a silly question, but with having the one ASO that has already been created, when you create another ASO for kids that weren't eligible for that one, is it a quicker process, or is it starting all over? Or are you pulling that one and just changing it a little bit?
It is pretty much starting all over because it is for a different SNP. The only thing where it would go faster is if, when we did the original screen, if there was a SNP that is then in common with like another one, that is then in common with some of the other kids, we could go back to some of those ASOs that were designed and see, how they did in the cells and like the in vitro assays to see how they did from a safety perspective. And then we could take those forward faster, but we still want to be able to screen at least, 500 if we can. Because if you screen that large amount of number, by the time you get to one, you have the best one. So
essentially it's all new.
It's an all new, but we're learning from the others. And if we have common SNPs, we will keep those on file and kind of like push those forward faster.
Thank you.
Yeah.
One last online question asks, are you considering other delivery methods for the ASO, such as intranasal or any other?
We will have to wait for the Ionis's and Dynes and Avidities of this world to develop those methods. So these being investigational n-of-1s, we go with routes of administrations that have been tried and have approved commercial trials. So for the brain, it is intrathecal. And I just want to also mention here, a lot of patients come or families come and say, what about doing a port? You know, you can administer through a port. Yes, you can, but not the ASO. The pharmacokinetics, the pharmacodynamics are known and understood for bolus push, right? So you push the entire drug over 1 to 3 minutes. It distributes. That's what we know. That's the 30 years of experience that we have, right? So ports are not possible or not going to be done right now. Intranasal, I would love that. So I just hope that my commercial colleagues get it done. And yeah, so it's intrathecal for diseases that are systemic, it's subcutaneous, and then for diseases of the eye, it's intravitreal. So those are the 3 routes of administration that we use.
Hi, so I'm Canadian and I found a physician in Canada willing to apply as well as do the procedure treatment. He actually has experience bringing ASOs into Canada for use. Would children in Canada need to be eligible for any current ASO, or could they apply for a new one? And if so, who would I connect him with?
So connect him with, just reach out to nlorem. I mean, you can also have him connect with me, the physician, and then I think we can move forward and see, is this an ASO that's already on the shelf that we can give? That's one path. Would these patients have SNPs that the other patients have, so that we develop an ASO together for multiple patients? That would be the other path. And so I would say have them reach out to us, and then we can go from there to get into the details.
Okay, thank you.
Yeah.
All right, thank you so much, Laurie. I think we'll close it out now and take a 15-minute break, but let's give a big round of applause.